Go. Okay. So my name is Bianca Perla. I'm the Vashon Nature Center Science Director, and I love this program of Salmon Watchers. I always get super excited. It was one of our founding programs, and it started before Vashon Nature Center even started. It's been going for a long time, so it's kind of an island tradition, and it's a fun one. I also want to introduce you to Alex Brown. Alex, can you give a wave so people can see you? Alex is our research and programs coordinator, and he's going to be handling a lot of the data management. So if you don't upload your data in a timely manner, he's the one that's going to go after you. Um, and then Kelly Keenan is also our volunteer coordinator. Can you give a wave, Kelly? Yeah, Kelly's up there. And she has been doing this for us for a long time. She has an amazing, uncanny ability to find fish and smell fish from everywhere. And she's been the volunteer coordinator for this program for a long time. So you will be getting the emails from Kelly weekly that update you on what people are seeing. And she's your first contact for any questions as you're going through this. Okay, so I'm going to start sharing my screen um, and get the slideshow. All right. Does everybody see that okay? Give me a thumbs up. Okay, great. So if there's too many other people that are in the view of the slideshow, you can go over to your little view panel where everybody has their faces, and you can hover over it, and there's a little grid on the top, and you can choose just show small active speaker window, and that will assign that'll just show whoever's speaking. Should be showing me, but it's not for me. So, but that'll give you a little bit more room on the screen to see what we've got here in the slideshow. So welcome to the Salmon Watcher Training 2025. I'm Bianca, as I said before. We've also got Alex Brown here and Kelly Keenan, and we're your team. And you are part of an incredible group on the island that has been doing the salmon watching for a long time. A lot of people have participated off and on through the years, and we also have our oldest salmon watcher this year is 99, Yvonne Cooperberg, and she's been watching salmon since the very start of when people started doing this on Vashon over 20 years ago, before our time as the Vashon Nature Center. We're just the current coordinator of this program. So this is a long legacy of people going out to the creeks and watching the salmon come home. So welcome to being part of it. It's a great experience. Why is this not? There we go. So the salmon watching exists as only one piece of Vashon Nature Center's whole salmon conservation monitoring and education program. So here's a look at the grand scheme and where you guys fit in all of this. Up here we have salmon watchers and trout trackers. You are the people that are keeping tabs on how many salmon and what types and where are coming back to spawn in island creeks. And so we're getting a log of that we can compare over many, many years and between creeks and between species of salmon. And Alex will show you a little bit about what we can find out about that. When the salmon spawn, they make little redds in the creeks. Redds are what we call salmon nests. Those are nests in the gravel, and we'll learn a little bit about that today as well, and how to identify those. And then in about March, or by about March or April, you get little baby salmon coming out. So we have a juvenile salmon inventory that we do with the high school students. So they're teaming up to have that next step in the life cycle and monitor that. So we monitor how many juvenile salmon are coming out of these redds and what species they are. We also have an exciting environmental DNA study that we're going to start this year, and that will be looking at this new phenomenon called non-natal rearing. Basically, we're finding out that salmon in the Puget Sound, especially Chinook and coho, are being born in larger river systems, but not rearing in the estuaries of those larger river systems like we thought that they did, as much as we thought. Some of them are leaving and traveling in the just first maybe inch or two top of the Puget Sound that's less saline, and they travel across and they find another creek to rear in. So they actually go across the Puget Sound, find a small creek that's in good condition, and do the rest of their rearing and growing there. And so we're going to be looking at our creeks on Vashon this spring, and because we're wondering whether juvenile Chinook salmon are doing that, if they're coming across the Puget Sound and rearing in our creeks. We don't have current records of Chinook spawning, adult Chinook spawning in our creeks, but there is a possibility that they're using our creeks for rearing. So we're going to try to get to the bottom of that this spring. So if you would like to be involved in that effort, keep your eye out. We'll have some opportunities for volunteers to help us with that. We also look at the marine environment, especially kelp beds that are really important rearing habitats for salmon. And so we're keeping track of how our kelp beds are doing on the island. We also look at shoreline health. There's several restoration sites where bulkheads have been pulled out in King County natural areas, and we look at how those places are bouncing back and whether they're providing the fish habitat that was hoped for. And so far, the answer looks like yes, they do. We also look at forage fish, which are little food for salmon, little fish that are food for salmon. We look at those and how they use the shoreline as well. We also do salmon education with the fifth graders and high schoolers. And we also have a stream team where we're just looking at the condition of the creeks. We look at water quality, we do water quality sampling, and we also do physical characteristics of the creeks, looking at the length, the wetted width, the depths, the temperatures, and we also collect invertebrates to get what's called an index of biotic integrity to look at the health of our creeks over time. So we have all these different things that all kind of mash together and create a really good picture for us to help us know what we need to do as stewards of these creeks, shorelines, and marine areas for salmon. And you guys are part of this big effort. So thank you. Because of the data that we've been collecting, we're starting to get a really good picture. We're starting to form a picture of how Vashon provides a unique function for salmon in the Puget Sound. And it has something to do with our geography and also with the good conditions of our shorelines and our creeks in comparison to a lot of urban areas that are surrounding us. So if you look, this is a whole aerial photo of the Puget Sound. Here's where Vashon is. And you can see that we really have this kind of central location, right? You know, we're an island right in the middle of the water, right? If you think about it from a fish's perspective, a lot of the big river mouths are emptying out just right across the water from us. You have the Puyallup River, you have the Nisqually River from the south, you have several rivers on the Kitsap Peninsula, as well as we've gotten fish from as far away as the Stillaguamish River up near Whidbey Island coming down into the Puget Sound, going around and foraging on our shorelines instead of going straight out to sea through the Strait of Juan de Fuca. So our island is kind of a magnet out there in the Puget Sound, providing this shoreline habitat and foraging habitat through our kelp beds and our beaches. And now we're wondering also our small pocket estuaries and our streams for these coho and Chinook that we're starting to realize are actually traveling over the Puget Sound to get to these areas. we're kind of a nursery. We're a central location. We're a nursery. The other thing that's interesting about Vashon is that our streams are all fed by our aquifer, right? We have a sole source aquifer, meaning that all of our water on the island comes from rain. It comes from the rain that falls on the island, and our streams are basically springs. They're basically groundwater that's coming out of our aquifer, the perennial or all year round streams. And then we have runoff from rains as well in the winter. But we aren't dependent on snowmelt like a lot of the bigger systems are. And because we're spring fed, our temperatures are a lot more stable. So one thinking that we have is that as climate change progresses, our creeks might become more and more important as cold water, steady refugia for these fish. And especially if that pattern that is just starting to evolve we're just starting to uncover in the Puget Sound, where we have little juvenile fish crossing the sound and rearing in these little creeks, these might become more and more important as climate change progresses. So all of your data is helping us paint this picture of how do we fit in terms of how do salmon see us in the Puget Sound. So I'm going to turn it over now to Alex, and he's going to do a little bit of more of what we are finding specifically with our salmon watching data. And then we'll come back and we'll run through how do you ID salmon and which types are in our creeks and how do you tell them apart and what do you do when you go out for your survey. And then we'll open it up to questions after that. So I'm going to stop sharing, and Alex, why don't you go ahead?
Okay, thanks, Bianca, for that kind introduction. And thank all of you for volunteering this season to watch our creeks. Let me share my screen. Slideshow. Okay. Can you see
Looks
that?
good. Mm-hmm.
Great. All right. So I know some of you, and I definitely recognize other faces. And I've read many of your names on spreadsheets with prior year's salmon watching data. And I've actually analyzed that data for a paper I wrote in a graduate course. So I'd just like to thank all of you for helping me with my education. And I even passed the course. So that's wonderful. Thank you. I volunteered as a salmon watcher for several years, and it's definitely one of my favorite citizen science programs that the Nature Center helps with. And I'll continue watching a reach in Upper Judd this season, so I'm definitely looking forward to it. So this is just a visual of the creeks that have been surveyed for the past 10 years. Not all of them are surveyed every year, but Shingle Mill and Judd are. And they are the two largest creeks and provide us with the most data that we get. So for the past 9 years, we have some stats here. They're always fun to look at. And before I go further, I should mention that I have borrowed some of the themed slides from Taylor's talk last year. And so she did a great job with it. And so I thank her for getting some really cool graphics on this slide specifically. But let's look at the specifics. So of the species, we had 625 chum, 432 coho, and 103 cutthroat. This slide is not exactly to scale, so that cutthroat will definitely be smaller if you see one in the creek. But it does represent that the chum is usually larger than the coho. So let's look at those two largest creeks, Shingle Mill and Judd. So the first thing that's pretty interesting is looking at the odd years. So odd years are when pink salmon run in the Sound. And studies have found that they usually compete with other salmon. And so you might see declines in those other salmon. And if you look at these too, you can kind of see some declines that happen in those odd years. 2020 is a bit of a strange year with COVID, and we had fewer people out looking at the creeks. But for the most part, especially in Judd, we see some declines in those odd years. Other things that we can see here is that 2022 was a pretty big year, especially for chum. And but then it dropped off in 2023. And so with 2024, you can see that there was a rebound for shingle mill with coho, not with chum, and then the opposite in Judd. The chum seemed to rebound a bit, but the coho actually continued to drop off. So this year is going to be really interesting to see what we can get. It's an odd year, so that may impact what we see, and also just to see where this trend will go. Yeah. All right. So focusing in on shingle mill, these are the reaches that are being observed. And you can see that, you can see that the chum prefer the lower reaches, or not, prefer is the wrong word. Chum are observed in lower reaches. And part of this is probably because they are larger. They're a little bit less agile than coho, slower if you see them in the creek. And so they tend to be in the lower reaches. And you can see the coho have a greater range. They're found, have been found, the farthest sightings of coho have been up to Needle Creek, and also branching off on the main stem of Shingle Mill, almost up around where West Side Water is. And so that's pretty cool that they can get up that far, because it gets pretty steep after you leave reach 5, between reach 5 and reach 6 there on the map. The gradient definitely increases, but coho have been spotted up there around Needle Creek. All right. So then let's look at the reds in Shingle Mill. And as Bianca was saying, the reds are these nests that are dug in the stream bed, and it kind of follows the same pattern as where chum and coho are found. in that that's where they will dig their nests. It's interesting to see that Reach 4 has the largest average reds found, and part of that might be that the substrate in that reach is really good for it. It could be that the gradient is good, or the combination of those two leads to many riffles that are great for reds to be dug in. But generally, coho will swim further up the reach, up the creek. And one of those reasons is also just because they don't have to compete with the chum that are digging their reds in the lower reaches. They can go further. All right, now switching over to Judd Creek, we'll look at their salmonid distribution. And again, it's similar to Shingle Mill, where chum are found predominantly in the lower reaches. Although the farthest sighting of a chum was in Reach 7, which is up by Jesus Barn. And that was in 2022 when there was high water, some big rainfall that allowed it to get up that far. And coho reds, sorry, not reds, coho have been found up past 204th on that reach, which is pretty impressive as well. All right, so looking at the red distribution, on Judd Creek, it's pretty even for coho, but the chum again will be found in those lower reaches. Although you'll see in the graph, the bar graph, that the chum that were found up in Reach 7, they had also dug a red. So there was a pair there that were spawning. So that's pretty cool. Okay, and here's something a little different data that we have that you might be interested in. So this DNA work, this comes from the fin clips that you guys take when you find carcasses of these salmon in the creeks. And this is what we get after we send those fin clips to WDFW. They analyze them and provide us with this data. And so these are 6 samples of coho. Each column is a coho, and the percent that it matches a population in those in the left columns. And so you can see that there aren't really any 100% matches, which we do find with chum fin clips. The coho database that WDFW has is not as well refined as the chum one, so it's harder to get an exact match, or they don't have a lot of the ability to do that yet. It's still being developed. So of these 6 coho, the 5th column one is from Judd, the one that ends in 19. and the others are all from shingle mill. And you can see that it's kind of a lot of the coho just have a mix of different populations. And so, this doesn't necessarily mean that we don't have a unique population to Vashon, because that population, it would take many generations for that unique genetic marker to show up. And so, as Bianca was saying, with the new studies of looking at juveniles that are exploring different creeks, it gives you a new idea of what a stray is. It's usually, we thought of strays as being adults that return, and for whatever reason they end up in a different creek than their natal creek. But now it could be that they are straying when they're juveniles and possibly getting imprinted with the creek they're exploring and going back to that one and not their natal creek. So there's still a lot of work to be done on looking at this DNA, but we definitely appreciate getting those fin clips from you guys. And the more we get, the more we'll be able to learn about this stuff. And here's an idea of that DNA work on the chum fin clips that we've sent in. And so this is just kind of giving you a visual of where those chum have the populations that they've been linked back to. And it's pretty big range there. And as I said, some of these chum, they will come back saying this is 100% from Minter Creek. But yeah, these are 14 samples altogether, and 6 from Judd, 7 from Shingle Mill, and one of the samples that we sent in was actually from KBI. And that one was linked to Grover's Creek. So it's pretty interesting stuff working with this DNA, and we're hoping that we can get more fin clips. It's tough because we are competing with raccoons and otters and coyotes, who will snatch up those carcasses pretty quickly if they are just lying there on the banks. So it's always great when we can get them. So the takeaways from what we've looked at: so different creeks have different characteristics. Fin clips are important, so we'll try to keep collecting those, and then definitely the eDNA work is going to be really interesting to find out what that can tell us about what's in our creeks. And also long-term data is important. It can help us look for patterns, things that are going on. And it can also show us, the longer the data set, can see if things are just a short-term blip, or if really things are changing in the long term. And all this information is helpful for the land trust, which has done a lot of restoration work in both, well, certainly both Shingle Mill and Judd, and probably other creeks that are being surveyed. And King County also uses the information that we collect to make decisions on places to help out to restore, and probably where to put funding. So that's all I have. I will give you back to Bianca. Thank you.
Thanks, Alex. If anybody has questions, you can pop them in the chat, or we can do questions at the end so that we're progressing through and getting through all of the protocols and stuff. So if you think you're going to forget your question, pop it in the chat. That's fine to do, and we'll address it later. All right, so I'm going to share again, pick up where we left off. Okay. So now we're going to go to your part, and what you're going to be doing out there. You're helping us answer 3 major questions. The first question that we want to know is which salmonids are using which island creeks and when. So when are the chum coming up, and in which creeks, and when do they start and when do they end? And when are the coho starting to run, and which creeks are they running in this year, and when do they stop? And so people who are assigned sit spots, that's what a lot of your data goes to, is looking at those large distributions and timing of our runs. We also have salmon reach walks, and people who do the reach walks will walk a section of the stream, and they will count fish numbers, and they will also count spawning effort by counting and identifying reds. And that's a quantitative measure of how much spawning is happening in our creeks. And so that's the second question. And then both reach walkers and salmon sit spot people, and just people who are out on the creeks. So your friends who might not be in this program, but might be going and looking at the creeks once in a while, tell them if they come up with a dead fish, we want to take a little fin clip off of it and get some DNA. So everybody's going to be looking for dead fish, no matter what you're doing. And that will help us. That's already, like Alex said, helped us paint this picture of our island being this center of exploratory runs, lots of diversity in our genetic of our runs here, which is pretty interesting. So having more information about that will be really nice to have. So what we're going to do now is we're going to go through key identification features for each of the fish that you may see in our creeks. And so in order to do that, there's some things to keep in mind about the fish that I will be talking about when I talk about identification. One is the gill cover. That's this little kind of the cheek of the fish. Another thing is the lateral line, which you can see goes right down the middle of the fish there. It's actually a sensory organ that allows them to sense pressure. It's one of the things that can help fish school, because they can sense what's around them better than we can. Dorsal fin is on the top. Another important thing is an adipose fin. It's a little tiny fin on the top of the fish, on the dorsal side of the fish, that's between the dorsal fin and the caudal fin, or the tail fin. And that fin, not always, but most of the time, is clipped in hatchery fish. So if you see a fish, if you can get, it's hard to tell, but if you can get a good look, one of the first things that's good to look for is, can I see the adipose fin or not? Because then we will know, we'll have more of an idea of whether it's a wild fish or a hatchery fish that's found our creeks. And then the caudal fin is the tail fin. That's what allows that's just the powerhouse of the fish. And there's some identification marks there that we want to keep track of. The other fins are not as important in terms of identification within salmon. So the questions to ask when you're identifying a salmon, because you're doing this in the field and sometimes you just see it very fast, ask yourself, what size is it? Does it have spots? And if it does, where are the spots? What is the color of the fish? And how is it behaving? So those are the 4 things that can really nail down what the differences in the different salmon that we see in our creeks when they're spawning. So here is a good size comparison. Chinook is going to be the biggest fish. On the left here, I have in red the fish that have been confirmed on Vashon in the salmon watching program as spawning in our creeks. So chum, coho, and cutthroat all spawn in our creeks. We know that. We expect them to be here. In odd years like this, we have seen stray pinks come in. So it's important to also know what pink salmon look like, especially this year, because they might come into our creeks. We haven't seen them spawning, but we have a few records of them wandering in, and so it's important to know what they look like. Chinook, we have there is one historical record in the salmon database at WDFW for a Chinook in Judd Creek, but we haven't seen any Chinook spawning currently. So that's another one to keep an eye out for, just in case it happens again. So we'll let you know what those look like. Sockeye and kokanee are not on Vashon. They are both species of salmon that need freshwater lakes as part of their life cycle, and we just don't have a freshwater lake that is, Fisher Pond just doesn't cut it for them, and can't get to it anyway through the water systems here. So we don't have sockeye or kokanee in our streams. So looking at the size range, you have Chinook is the biggest, chum is the second biggest, and then coho, pink, and cutthroat. Most of the time you're going to see cutthroat. It says up to 30 inches there, but you're going to see mostly cutthroat that are about 6 inches or less, because they're coming up to eat the salmon eggs at this time. Spots. Chinook will have large splotchy spots on their dorsal side, and they will also have large splotchy spots on both the top and the bottom of their tail. That's important because coho are the other spotted fish, and they have small, distinct spots that only appear on the top half of their tail, not the bottom. So if you're seeing a big spotted fish in our creek, and you think it might be a Chinook, see if you can get a look at the tail, because the tail will tell you for sure whether you've got a Chinook or a coho. If you only see the spots on the top edge of the tail, it's coho, and it's just might be a big one. Pink have large oval spots as well, and they have spots all over their tail, not just on the edges. And then cutthroat will have numerous small spots over their entire body, not just above the lateral line like they are on the other salmon. Color. Chinook can be a wide range of color from red to green. They can look a lot like coho. Chum are really distinctive. I think of them like 80s race cars, like Trans Ams or something. They have these like flame markings all over them, and that's very reddish and purple flame markings and no spots. So they're very distinctive. You'll know when you see a chum. Coho. Oh, the other thing with chum is the females and the subdominant males will have a thick black line sometimes that runs right down their lateral line, and that's just kind of a line that they can actually turn on and turn off, but you might see that it's kind of a sign of stress, and it's also a sign of, hey, I'm not dominant. It's sort of a sign of I'm not a dominant fish. Don't bother with me. And it's a big thick black line, and I'll show you some pictures of that later. Coho have red on their belly and their gill covers. Kelly has a good saying. She thinks of coho ho ho. Ho ho like Santa with kind of ruddy cheeks, and that's what coho have. So you'll see, especially later in the season, that red forming. If you see a gray fish with spots, it could also be a coho that just has come in from the marine environment and it hasn't fully changed into its spawning colors, or females tend to not be as red as the males. Okay. So here's a review. Coho have these round black spots that are fairly small on their dorsals and the top part of the upper lobe of their tail fin. And then their lower sides can be red, purple, and they can have kind of blushy cheeks. The other thing about coho is as Alex said, they're really acrobatic. So they can get way up high in our creeks because they are really good at swimming in hardly any water, and they're really good at jumping over logs and other obstacles. So if you're seeing a fish that's really fast and it's jumping and it looks really skittish and acrobatic, that's another sign that you might be looking at a coho. They also splash a lot when they're trying to get up over things. So you might hear them before you see them. So here is a picture of a coho in a shallow stream like ours. You will see them half out of the water like this when they're coming up, which is pretty amazing that they can get up these places. The chum, also called dog or Kita salmon, have really well-developed teeth. Some people say that's why they're called dog salmon. Some other people say they're called dog salmon because people feed them to their dogs because they're not as good tasting. But chum have these really big teeth. So if you're seeing a carcass or something and it has really, really big teeth, it's probably a chum if you can't see any other identifying marks. But it has these flame black, it doesn't have any black spots, and it has these flame markings. Dark blue, reddish purple, brownish, depending. These guys' behavior, they're calm, they're slow, and they're bumbly kind of. I watched for about an hour, I watched a chum try to get up over a log on Judd one year, and it was so frustrating to watch because it just couldn't do it, and it finally turned around and went back. So these guys are not as acrobatic, and they're big and kind of thick-bodied compared to the coho. So that's another thing to look at. Here you have a picture that shows their teeth and their flame markings really well. Down on the bottom, you can see that black line that I'm talking about. So that's a sign of either a female or a male that is not dominant and is trying to look like a female so he can sneak in on the mating without getting hurt. So you'll see that. If you see that black line, it's definitely a chum. There's no other fish that does that. Chinook, you can see here spots on both the upper and the lower lobe of their tail. So that's very important to get a look at if you're wondering if you're seeing a Chinook. If you think you're seeing a Chinook, these guys are big. They're bigger than chum. And they're calm, but they're really powerful. So they're kind of a mix of that coho acrobatic and the calmness of the chum. But if you think you're seeing one, call Kelly. She can come out with her GoPro and confirm with you. Here's another look at the patterns of the splotchy sort of big spots that you see on the dorsal side of the Chinook. And you can see here also on the bottom, that's the size of a Chinook, and then those other fish in the background are coho. So you can see how much farther it's sticking up out of the water than those big, big fish. Cutthroat trout, you can see where the spots are everywhere. These guys are little. They have kind of more little button noses. They're super cute. They are the fish of our creeks. They're doing really well in our creeks. They are going to be much smaller, 6 to 8 inches probably. They come up a lot of this time. They live in salt and fresh water, and unlike the salmon, they can come up, spawn, and leave. They don't die. They can spawn multiple times before they die. but they're not spawning usually right now. So they're coming up into our creeks specifically to eat the eggs that the salmon are depositing. And so you might see them flitting around. And we also have small resident trout in our creeks that you will probably see doing the same thing. So cutthroat trout start spawning in January or February. And we sometimes have a trout trackers program. So if you want to keep going to your spot and start looking for trout after the salmon are all gone, you're welcome to do that too. This year we need to keep an eye out for pink salmon. You can see, if you see a male pink salmon, you'll know you're seeing a male pink salmon. Look at that huge hump. So they're also called humpies. And you can see how their tail is, it just has these huge black neat spots all over it. That's really a typical sign that you're seeing a pink. And then they have sort of farther apart spots on their dorsal side than coho, bigger spots than coho, probably the same size spots as Chinook, but they're farther apart than the Chinook. So they also have kind of a greenish color on their back. They kind of shine a little greener than the other fish. That's coho. And they're fairly small. You can see the size compared to that person's boot there. I don't know if you guys remember, but a long time ago, 2017, there was that net pen spill of Atlantic salmon that led to this not being allowed in Puget Sound anymore. But it's always good to know what Atlantic salmon look like, just in case some of them survived. For a few years, people were getting Atlantic salmon in the bigger rivers like the Skagit. Seems like haven't really held on. So that's good because they aren't native to this area and they could compete for salmon spawning habitat. So the one thing to look at is if you see a fish that has those spots right on their gills, but not really anywhere else, right on the gill cover, that could be an Atlantic salmon. And so make sure to take a picture of that and let us know. Here they are in spawning colors, and you can see they still have spots on their gill covers, but they kind of start to turn cool colors. They also look sort of more amber colored than our other fish, than our native fish. All right, so a few comparisons just for review. Difference between coho and chum: spots, flame markings, red belly and gill cover, and sharp teeth for the chum. Coho tend to come up a little sooner than the chum, and that's because they can get up in lower water conditions. So the chum usually, we call them our Thanksgiving fish because they usually start arriving end of October, early November when the rains really start in earnest, and that opens the creek up to them because it makes the creeks deeper and more easy to navigate. Coho can navigate better, so they tend to come up first, but not always. They're skittish, they're acrobatic, they're thinner in their body, and they can go very high in the watersheds. Chum, slow, thick body, spawn in the lower parts of our watersheds. Difference between Chinook and coho: large size difference usually. They have large splotchy spots, whereas coho have small distinct spots. The big thing is spots on the top and the bottom of the tail for Chinook, where you only have it on the top half of the tail for coho. They both can be red. Color's not really a good distinguishing factor between Chinook and coho. And they both spawn at the same time. Chinook tend to be a little calmer and less skittish than coho, less shy. So I just want to let you know that we're going to give you some Salmon Watcher resource packets with in a couple. Kelly will email them soon before we start, and you will have a link to this presentation, and you'll also have a link to a PDF that has all of those pictures and all of those identification characteristics, and it also has a quiz that you can quiz yourself on. So that you'll get that, and you can just load it onto your phone and keep it with you, and you can check back with it in the field. And if you have any other questions about salmon identification, you can always text or email any of us, or ask us what you want. The next thing I'm going to talk about really quickly is how to identify a red. So we're talking about how these salmon will deposit their eggs in the gravels. What happens is the female will come up and she will excavate the gravels. So she will flip over a lot of the rocks and make a little pit that she can use to put her eggs in. And so in our creeks, this is a very big red in a large river, and it's actually probably a collection of reds right next to each other. But you can see that in a creek, the rocks get a kind of a patina of algae, and when they get turned over by the fish, you get this lighter colored rock. So all in that picture, all along here where it's light colored rock, that's all what the female, probably in this case, Chinook, just looking at the size of the gravels, or maybe chum, have excavated out. And so the reds are in here. That will eventually fade, and these will get a patina again. So that's why we go out every week, is because we can see then those fresh reds, and they fade in about a week. Reds are usually to the side or just upstream or downstream of a riffle. A riffle is that sort of fast-moving water in the creek. And the reason they put it there is so that these eggs can get oxygen from that moving water, but they don't get taken out by being right in the middle of a riffle. So to the side or just upstream, near a pool or other bank cover like logs or overhanging trees where the adult female can hide. The females will come up, they will make their reds, they will deposit their eggs and mate with the male, who will fertilize the eggs in the gravel. The males will continue on and try to find more females. The females will stay with their reds, and they might make a few other reds next to their first one if more males come up, but they'll stay in that area and sort of guard their reds for the rest of the season until they die. And so if you're seeing a fish that's sticking around, they probably have a red there. So this is just a diagram of what a red looks like and where the eggs would be. So you can see that they kind of excavate out this pit, and they put their eggs in that upstream part of the pit. The fresh reds will be lighter colored than the other ones. So that's how you can identify them. And Kelly and Alex will go through this in the field for anybody who goes to the in-person training on Sunday. So when you get out there, what do? When do we start watching? We start watching September 30th. But if you have your spot and you want to go out earlier, go for it, because sometimes the fish come up earlier. We stop watching December 31st. How often do we watch sit spots? You will watch twice a week for 15 minutes or so. If you want to, you can also, just do once a week for a little longer. Reach walks once a week. You will walk your reach on the stream. We watch from the side of the stream as much as we can so that we're not disturbing those reds with our feet. Sit spots, you're looking for the number and the identity of the salmon swimming by. What species is it and how many? And the reach walks, you're looking for what species is it, how many are you seeing, and then also how many reds are you seeing, and are you recording new ones each time? Every time you see a red, if you're doing a reach walk, you will mark it by tying a little bit of flagging to a nearby tree or overhanging branch. And so the next time you come, okay, I already marked that red, so this must be a new one that I didn't mark. Everybody look for those dead fish and get a clip the reach walkers will actually, because they have more of a chance of seeing dead fish because they're going farther along the creek, you'll get a little packet, a little kit that has scissors or a knife in there that you can get a little bit of tissue from the fin. We'll also have a few wild cams out there that Alex and me and Kelly will be manning, and we'll share what we find with those because those are kind of exciting to see what's coming to the fish and what's coming to investigate the stream at this time when there's so much bounty from the sea coming back. So what should you bring? Something to record your data with. Usually it's a phone. We have, I'm going to go through the data forms. Any salmon ID materials like the cheat sheet that we give you. Your phone camera or a different camera is good. Polarized sunglasses sometimes help you see a little bit better because they cut down on the sunlight glare on the water. We don't have paper data sheets anymore, so we don't do clipboards, but you can do an orange vest if you want, if you're worried about hunters at this time. Most of the places on Judd and Shingle Mill anyway, where we're looking, wouldn't have too many hunters, but it is hunting season. So if you're in a spot where you think there might be hunters, you can wear an orange vest. Flagging and permanent marker for the salmon reach walks and knife for fin clips. And you can also bring a buddy if you want. That's fun for safety and also for sharing your experience. Okay, so I'm going to go to entering the data. You're going to get a form that looks somewhat like this. Can everybody see that okay? This is a Salmonwatch. This is for the sit spots, and we'll just go through this one because they're pretty similar. This is where you're going to be entering your data, and we would like you to enter it each week. So you're going to be going to the creek each week, and we want to have your data there. You can put your name. I'll just do it one. Let's say I'm doing Shingle Mill. Each of you will be assigned a reach. Maybe I got assigned this sit spot number 1. You put your date. It's the 9/27. I can't remember what it is today. And then you're going to do a start time and an end time. And these are important because we then can calculate effort. How long have people gone out there for? And then, so this is new this year, and some of you who have done this before will love this. Did you spot any live or dead salmon? A lot of the times you won't. And so if you put no, you're just going to have one more question. So I'll do that. Did you see any juvenile fish? Any little things that are 1 to 6 inches? Maybe I saw 2 of those. And then you have a time to say any additional notes that you wanted to share with us. If you did see a live salmon, it'll take you to a longer form to fill out. So how many coho did you see? Maybe I saw 2 live coho. And how many dead? None. And how many live chum did I see? Maybe I saw 1 and 1 dead one. This would be an amazing day, by the way. When Then we want to know if you are seeing any salmon, was it clearly missing its adipose fin? So remember that little fin between the dorsal and the caudal fin. If you noticed and it did look like it had fins, you don't have to say anything. But if you did see one that is missing, you could say one of the coho had no adipose fin. Or you could say, I couldn't see whether the fish had an adipose. You can put whatever you wanted to put there to let us know about that. And then asking trout, The last thing is if you do see a fish, but you really can't tell, because sometimes you're just going to get a really quick view of it. If you can't tell what it is, just err on the side of caution and say, you know what, I did see a fish, but I don't know what it is. Just say, the water was too deep. I couldn't see markings or something. Oh, or, you know, might have been a chum, but not sure. So you can write whatever you want in there. And then next, it'll ask you if you want to submit. I'm not, because I don't want to screw up Alex here, get him too excited that we have fish. So I'm just going to clear it for now. But that's how you do it. It's just a Google form, and you can do it on your phone. Right after, I usually do it when I get back to the car. But you can do it on the phone, or you can wait until you get home and do it on the computer. Either way. So, I think that's all we've got. Important tips, and Kelly will give you this in her next email. You can call Kelly with any questions. You can email her with any questions. Take lots of photos, even if it's stuff that's not fish. We were able, you know, some of our salmon watchers found the first record of American dippers in Judd Creek. There's these little beautiful water songbirds, that are pretty rare in our island creeks. So anything that you think is cool, please send. You guys are the eyes and the ears on the creeks right now. And even if you're not seeing salmon, there's lots of other stuff to see. Please stay on public property unless you have permission from the property owner for your site, or you are the property owner. And then while you're out there, you now know a lot more about salmon than the most people just after hearing this talk. And so feel free to share. Share your knowledge, share your enthusiasm, share encourage correct behavior. If you see somebody's dog going into the creek right around some reds, just tell people nicely, you know, hey, I don't know if you noticed, but there are a lot of salmon reds there, and your dog could disturb it. So could you kindly take them out of the stream or something like that? So you're kind of ambassadors while you're out there too. So if you haven't gotten a site already, Kelly will work with you to assign you a site. Most of you, I think, have signed up on our events page. You've signed our form, which included the waiver. So we're at the step where Kelly will work to get your site. We'll make sure that you can access those online data forms and the volunteer manual. Those will be coming soon to you. Write down anything you need, any maps or directions or phone numbers. If you're really not feeling like you can figure out where this place is, just ask Kelly or Alex or me, and one of us can go and help you find it in person. Once you have a site and the link to the data portal and you've signed our waiver, you're all set. You can go out onto the creek and enjoy your spot. Couple things. Tell Kelly if you're going to be absent so she can find somebody to cover your spot. And also record your data each week so that Alex can stay on top of giving us the salmon counts each week in the email that Kelly will send out. And lastly, thank you for doing this. There is no way that the staff at Vashon Nature Center alone could do this without you guys, without all of the boots on the ground. We just could not cover this much of our creeks and see the salmon coming home in the way that we're able to with this whole network. So thanks for doing it. And I hope